All right, everyone, let's get back to it. So, back to spontaneous generation theory, because this idea, we're going to talk a lot more about it. You might think that now that we've covered cell theory, that was that obviously that idea was gone to the wind. But spontaneous generation theory actually persisted for a long time, even after cell theory really took shape. So, just as a review, spontaneous generation was this idea that living things could spontaneously arise from non living things. It was this early idea to explain the origin and appearance of life, why it was that things would just appear seemingly from nothing, why mice would appear from old hay piles, for example, that were rotting in someone's fields, or why maggots would seem to arise from rotting meat as it decayed, or why bread would start to turn blue and develop mold, this very distinctive mold, and many more. And so this was really something that was born out of our inability to witness these events take place. People were not able to see microscopic molds land as spores on the surface of bread. They were not able to see the tiny eggs being laid. People were not paying attention or were not literally able to witness these things. So again, limits of human perception taking place here. Now, over time, as science developed, people began to really target spontaneous generation as a thing that was starting to make less sense. That there were too many questions, and it was getting harder for people to rationalize the things that we observed over a long time as being as essentially being related to spontaneous generation and not some other explanation. And so some scientists began to devise experiments to actually test spontaneous generation. These people were starting to apply in these early days. It was very new, but the scientific method, which was still a relatively new tool at the time. So, as a quick review of what the scientific method is, it's a framework that we can use to come by reasonable conclusions to major questions that we have that are, to the best of our understanding, backed by evidence. And by that, we hope that means that they're closer to the truth, or as close to the truth as we can get. So the scientific method goes a bit like this. You start with a question. When you have your question, you do a bit of background research to learn about that study topic. And when you have a little bit of information that you can use to make predictions about it, you do that. You form a hypothesis. A hypothesis is really just a framework of prediction. It's when you say, I think that the outcome will be this because, and then provide some kind of evidence. That these are educated guesses, a realistic potential outcome that you think might happen. Not that you think will definitely happen. We want to make sure that we're not being, subjective when we or biased, for example, when we start our experiment. But we want to come up with ideas about what we think might happen. When we have those predictions, we can test them by creating a framework to do an experiment. Essentially, our goal is to evaluate if we were correct or incorrect with our hypothesis. And when we do that, we get our results, whatever those results are, and we draw a conclusion. That conclusion should really be, was my hypothesis supported by the evidence? Yes or no? Why or why not? And then finally, if need be, you report those findings and or formulate a new hypothesis. So if in the example of being incorrect, that your hypothesis was wrong, that doesn't mean the story ends there. you then also can create a new hypothesis for you to test. So it's very cyclical. You can go in a circle here until you get closer and closer and closer to the true story, to the best of our understanding. An example of this put into a very simple framework would be a question like, where did I leave my keys? That's my research question. And for background information, I can look back at my life and my history of where I have left my keys in the past. Or maybe I could look at where other people have left their keys in their lives. And so I can come up with a hypothesis. I could say, I think I left my keys in my car. I think I left my keys in my car because I've done that before. Then it's time to test my hypothesis. In this case, I'd have to go to my car and look for my keys. That's testing the hypothesis. If I found my keys there, I can say that my hypothesis was supported by the evidence. My keys were in my car. If I didn't find my keys there, then that means that my hypothesis was not supported by the evidence. My keys were not in my car. What now? Well, the story can end for me if I found my keys, but if I didn't, then I'm going to have to come up with a new hypothesis. Maybe I left my keys at my office, and then I'd have to go and test that hypothesis. Rinse, repeat until you find the true answer, or as close to true as we can realistically get in this world. So, let's take this idea and apply it to spontaneous generation, because people at the time were thinking exactly this. How would we test this idea of spontaneous generation? Spontaneous generation is the hypothesis in this example that life could arise from non-life. Then it should be testable. We could create an experiment to test that. So take a moment right now to think about what the structure of that experiment might look like for you, okay? And then we're going to go and talk about what other people throughout history actually did. And we'll see maybe how close you got. So the first one on our list is Francesco Redi's experiment. Redi was an early pioneer of a lot of important things in biology, but one of them was the idea that spontaneous generation was not supported by the evidence. He believed that life could only arise from life, which should sound very familiar to you right now. It should sound a bit like cell theory, one of the tenets in cell theory. So, this idea that cell theory purported that essentially that life could only arise from life, that predates cell theory by a long time, by many centuries. Redi is quoted as saying, "Omni vivum ex ovo," which means every living thing from an egg. His experiment in 1668 sought to actually demonstrate that spontaneous generation was not true. And so what he did is he created an experiment where he, in 3 jars, left meat out to spoil. He put a piece of meat in a container that was left open to the elements. He put a piece of meat in a container that was corked shut and sealed. And then he put a piece of meat in a jar that was sealed only with cheesecloth or gauze. And so it was porous. It was something that air could move through, which was a big thing people were concerned about. People thought that life could only arise from non-life if it was exposed to the air, for example. Lots of different ideas that people came up with to try to make that idea work. So Redi capitalized on that. He essentially said, I know what you're going to say. And so I made this experiment where the meat is still exposed to the air, but is still sealed. Still sealed from things to get in there, at least large things, macroscopic things that were not so small that could not be seen by the naked eye. And so he let these pieces of meat rot and spoil. And what he found was the container that was left open to the elements had maggots and flies that were on the meat. The piece that was left in the corked container, it did rot. It didn't bacteria were on it. He didn't know that, but it still spoiled. It wasn't good to eat. But there were no maggots and no flies, importantly. And then the container that had the cheesecloth on it, well, it similarly it rotted, but there were no maggots and flies on the meat itself. Instead, flies and maggots were found on the gauze, not on the meat, which indicated that maggots and flies had to enter the container. It had to be brought there from somewhere. They were not created by the meat, but had to be brought by something else, in this case, the fly. So this was a really simple experiment, but it was very effective. If flies were born from the rotting meat, why was it the case that no flies spawned in the sealed containers? It was an important question that proponents of spontaneous generation theory now had to contend with. And maggots that developed on top of the gauze lid, right, they grew there, indicating they were brought there from somewhere else. They were not created from the meat. It seems, from our perspective right now, that this experiment should have done the job, right? That this experiment should have put the final nail in the coffin of spontaneous generation. Well, no. So Reddy's work was met with a lot of skepticism, and a lot of people, because bias is very easy to come by, and it's very easy for people to become invested in ideas and be upset when they're proven wrong and things like that. Well, people didn't really like the idea that spontaneous generation was wrong. Or found new reasons to poke holes in Reddy's work. And so while his experiment today is viewed as a very strong one, very clear-cut test of spontaneous generation in which it failed, it would take another few centuries for people to finally abandon the idea of spontaneous generation, which means more experiments, more demonstrations like Reddy's. So I've got a couple that we're going to talk about. One of them is Micheli, Pierre Antonio Micheli. In 1729, he was very interested in fungi. So he took melon slices and he brushed the spores of fungi onto them, because at the time it was unknown that spores were the progenitor of fungi, that spores would grow up into fungi, basically. And so that needed to be proven. Before, it was believed that fungi would arise spontaneously from food as it broke down and spoiled. That it was something that was already there, that it was created by the melon in this case as it rotted, not a separate organism. And so Micheli found that the slices of melon that he put the fungi on, the spores on, well, that's where the fungi really grew. They didn't really grow on the ones that were devoid of spores. And so he demonstrated here that fungi were not something that generated spontaneously. Still wasn't the nail in the coffin. Later, Lazzaro Spallanzani in the 1760s did a different experiment where he boiled 2 different flasks of broth. One he left uncorked and open, and one, after boiling, he corked. So as soon as it stopped boiling, he corked it, and then took it off the flame and let it sit for a long time to see what would happen. And what he found was that only one of those broth flasks grew fetid and rotten. And it was the one that was exposed to the air where microbes existed, right? Now, Spallanzani, again, 1760s, had no idea about, much about microbial life, and certainly not if he was aware, he wasn't he didn't understand to the extent that they were influential. But what he did discover is that sterilization could be achieved through boiling. That you could essentially, again, he had no idea about microbes really, but you could extend the life of something by boiling it and preventing it from breaking down prematurely, from being spoiled early. These ideas continued to persist, and even as cell theory developed in the 1800s, people still clung to these ideas of spontaneous generation. Cell theory states that all cells, all forms of life, only arise from other pre-existing cells. You had to have cells to create more cells. And so this idea was something that directly competed with spontaneous generation at this time. In the 1850s, we can say that this idea was finally put to bed by Louis Pasteur. He's well known in the field of microbiology for a bunch of different things, but this is probably the first. And so he really helped put this idea to rest, the experiment in 1859, where Pasteur did a very similar experiment to Spallanzani. What he did is he had these flasks, these swan neck flasks, and he boiled both of them. And when he did, he took one of them and snapped the neck off of it, and for the other one left it with the neck on. So these swan neck flasks, they're really interesting because you see this long neck on the swan neck flask. When you boil this fluid that's in here, the escaping gas, you're boiling water, you're creating steam, right? And it rises and it goes into the neck of the flask, and then it begins to cool. It cools as it makes contact with colder air that is in the neck of the flask and the colder glass, and it begins to condense on the surface of the glass and run down. Eventually what happens is water will accumulate in the swan neck flask and essentially form a bit of a barrier where things can't easily go up through the outside of the flask, through the neck, and then into the flask itself. Particularly because there is now a barrier of water in here. So a lot of this stuff will get trapped down here in this water if anything goes through this the opening in this flask. In the experimental flask, Pasteur broke the neck off of this one, so it was just open to the environment. This one still remained sealed. And so what he found was that the swan neck flask that still had the neck intact, well, it for days remained clear. The broth was fine. There were no issues, and it didn't spoil. But over here, for this experimental one where he broke the neck off of it, that absolutely spoiled. It spoiled very quickly. And this was actually during this time where Pasteur had access to a microscope. He could put these under the microscope and look and see what's in them. And so Pasteur is also credited with helping forward the discovery of microbes as a spoiling agent for food. That he actually showed that microscopic organisms that could be killed with heat were present in the environment and could actively spoil food. So people used to believe that spoilage was something that was an inherent property of these foods, that living things would grow from the non-living or now dead materials of that food. Pasteur demonstrated through this experiment that was not the case. That environmental microbes getting into your food, those are what eventually drive it to spoil. So we can really thank Pasteur for advancing this idea of against spontaneous generation, and essentially like putting the final nail in the coffin of that idea. But we can also, at the same time, thank Pasteur for recognizing that microorganisms cause disease. He's actually responsible for the thing that we call germ theory, the idea that infectious disease is caused by infectious agents, that bacteria and viruses, things that people at the time called germs, we still call them germs today, but we know much more about them. We know what they look like and what they are and things like that, that they can infect a person or some other organism and cause a disease, cause the symptoms of a disease. That was a very new idea at the time. Like people had lots of other really wild ideas about what caused disease, and it was oftentimes a bit scattered about what caused disease. Pasteur, his work helped focus a lot of awareness towards the actual cause of infectious disease, microorganisms. Later, his rival, Robert Koch, these two hated each other, and they were fierce competitors in the field of microbiology. They were constantly trying to one-up each other. So that was simultaneously really good and really bad. But it did create a lot of really interesting and positive research in the process. Koch is responsible for a framework, a series of methods that people could use to help demonstrate that a specific disease was caused by a specific germ, a specific microorganism. These we call Koch's postulates. These are methods that we still use today in one way or another. So when we were dealing with the arrival of COVID, for example, and we couldn't figure out at the start what the causative agent of COVID was, researchers used a modified version of Koch's postulates to come to that conclusion. So these are still things that are utilized today to identify what is causing a specific disease. So Pasteur and Koch together were very influential and helped advance epidemiology, the study of infectious disease. But at the time, medicine had to play a lot of catch up because at this time, doctors are firm believers in something that we call miasma theory. This was the idea that disease was caused by bad air, not by microbial life, but by some non-living quality of air itself. Oftentimes, like the smell, or like the where the air comes from, things like that. This idea of miasma theory was eventually replaced by germ theory, but it took a while for medicine to really kind of catch up and realize that this was the case. It was something that happened as Pasteur and as Koch were doing their work. It was great, but before that, there were people like Ignaz Semmelweis. Semmelweis is an amazing character in medical history. So Semmelweis is responsible for essentially discovering that we needed to wash our hands in order to prevent the spread of disease. Semmelweis took over as the head of a hospital, and when he did, he realized that there were a lot of very unnecessary cases of infection, that there were women that were coming in to deliver babies, and a lot of these women would die, or otherwise become infected. They become infected and a lot of the time die during that childbirth. But he compared their mortality rates, the mortality rates of the hospital that he worked at, to a midwife clinic that was in the same city. And he found that their rates of this thing that we call puerperal fever, the childbed fever, childbed sickness that was killing women in his hospital, it was an incredibly uncommon cause of death if you went to go get delivered at this midwife clinic. A lot of doctors at the time didn't really pay that much mind, But Semmelweis took that very seriously, and he went on this long mission. He started a series of intensive experiments to try to identify what was causing the this high rate of puerperal fever, but more importantly, how to stop it, how to bring the rate down. What can he do to reduce the rate of that infection? Okay, so these he wanted to reduce these nosocomial infections. A nosocomial infection, by the way, is an infection that's acquired in the hospital. That was his main mission. Okay, so Semmelweis ultimately he identified this source. He realized that it was the sanitation practices at the hospital, that researchers, doctors were delivering babies after they had gone and performed autopsies, and they did not wash their hands between dissecting a dead person and delivering a child. And so they were bringing infectious particles. At the time, Semmelweis had no idea what that was. He didn't know what bacteria were, or what viruses were, or things like that. He was still thinking about this in the framework of miasma theory, but it made sense to him because he was observing real data, real evidence that if you washed your hands between treating patients, you could reduce the cases of these infections. So it was really impressive. Semmelweis ultimately, he couldn't convince the community, however, of his discovery. He didn't do a very good job of communicating this information, and he eventually lost his job and was replaced by another doctor who stopped all of these hand washing practices. So essentially, the rates of these nosocomial infections, the puerperal fever, it spiked again, and childbed fever became rampant for a long time. Semmelweis was almost forgotten by history until relatively recently, like in the last century, which is a good thing that he was realized, that he was rediscovered. Much later, some 25 years later, Joseph Lister became sort of the front man for the idea of sterilization, of sanitation, really, because he developed the first aseptic techniques, the first ways to sterilize tools and clean wounds that ultimately reduce these infection rates. Now, Semmelweis came before Lister, and kind of said similar things. They did come up with different solutions. Lister approached the situation from cleaning tools and cleaning like injury, like sites of injury, to remove bacteria. So he would like spray carboxylic acid on wounds to try to kill things that might be there that would cause infection, for example. Ultimately, together they were very influential. But this effort collectively was very important. That this was finally the application of Pasteur's germ theory directly into medicine, which was very important.